July 2018, Southwest Florida. Dead fish on the beach. No storm, no spill, no explanation, just fish. Hundreds of them, then thousands. The water was the wrong color, the air started to burn your throat, and whatever was causing it was invisible. This is the story of how a single-celled organism smaller than a human hair brought 150 kilometers of coastline to its knees. If this is your kind of content, like and subscribe. It's the only reason this channel keeps going. It started quietly. It didn't stay that way. By early July 2018, the surface water temperature along Florida's southwest Gulf Coast had climbed to approximately 30 degrees Celsius, or 86 degrees Fahrenheit. That warmth was not unusual for a Florida summer. What followed, however, was. The first reports came from recreational fishermen and shoreline residents near Charlotte and Lee counties. Dead fish appeared in isolated clusters along the shallow inshore waters. Mullet, catfish, and pinfish, species common to the estuaries and tidal flats of this coastline. No clear cause was identified at the initial point of observation. Marine monitoring data collected by the Florida Fish and Wildlife Conservation Commission showed something else in those same waters. Dissolved oxygen levels were dropping. In healthy coastal waters, dissolved oxygen typically registers between 6 and 9 milligrams per liter. In the affected zones, early measurements were falling below 4 milligrams per liter. At that level, most fish cannot sustain normal metabolic function. Oxygen depletion in shallow coastal waters is not automatically a sign of algal activity. Sewage discharge, urban runoff, and natural seasonal stratification can all reduce oxygen levels. In July 2018, investigators were not yet ruling out any of these causes. At the same time, water samples taken from the Charlotte Harbor estuary and surrounding nearshore zones revealed something more specific. Corinnia brevis, a single-celled marine dinoflagellate, was present in the water column. Cell counts at this stage were in the range of several thousand cells per liter. That range sits near the lower threshold for what scientists classify as a harmful algal bloom, defined by the National Oceanic and Atmospheric Administration as concentrations exceeding approximately 5,000 cells per liter. What made the situation more complex was that this bloom had not appeared suddenly in July. Offshore satellite-derived chlorophyll data and water sampling records indicated that a Corinnia brevis population had been present in the deeper Gulf waters since at least late 2017. That offshore population had remained relatively diffuse, too dispersed and too distant to threaten the coastline directly. By midsummer 2018, however, prevailing wind patterns and surface currents had begun to push that offshore biomass toward shore. The Gulf of Mexico's loop current and its associated eddies play a significant role in redistributing water masses along Florida's western shelf. In the summer of 2018, these circulation patterns, combined with sustained onshore winds, acted as a delivery mechanism. They transported the existing offshore bloom population toward the shallow, nutrient-rich waters of the coastal zone. Nutrient availability is central to understanding what happened next. In June and July 2018, the U.S. Army Corps of Engineers had been releasing large volumes of water from Lake Okeechobee northward through canals and also southward and westward via the Caloosahatchee River into the Gulf near Fort Myers. These releases carried elevated concentrations of nitrogen and phosphorus, the primary nutrients that fuel algal growth. Nitrogen levels in the Caloosahatchee discharge during this period were documented at concentrations several times higher than those found in offshore Gulf waters. What made the situation more complex was that this bloom had not appeared suddenly in July. Offshore satellite-derived chlorophyll data and water sampling records indicated that a Corinnia brevis population had been present in the deeper Gulf waters since at least late 2017. That offshore population had remained relatively diffuse, too dispersed and too distant to threaten the coastline directly. By midsummer 2018, however, prevailing wind patterns and surface currents had begun to push that offshore biomass toward shore. The Gulf of Mexico's loop current and its associated eddies play a significant role in redistributing water masses along Florida's western shelf. In the summer of 2018, these circulation patterns, combined with sustained onshore winds, acted as a delivery mechanism. They transported the existing offshore bloom population toward the shallow, nutrient-rich waters of the coastal zone. Nutrient availability is central to understanding what happened next. In June and July 2018, the U.S. Army Corps of Engineers had been releasing large volumes of water from Lake Okeechobee, northward through canals, and also southward and westward via the Caloosahatchee River into the Gulf near Fort Myers. These releases carried elevated concentrations of nitrogen and phosphorus, the primary nutrients that fuel algal growth. Nitrogen levels in the Caloosahatchee discharge during this period were documented at concentrations several times higher than those found in offshore Gulf waters. On August 1, 2018, the Florida Fish and Wildlife Conservation Commission confirmed what water sample data had been suggesting for weeks. Karenia brevis cell counts in the nearshore waters of Lee County had crossed the 1 million cells per liter threshold. That number is not simply a data point. It represents a biological state change, the moment at which an algal population transitions from a background presence into an active, self-sustaining ecological force. To understand what 1 million cells per liter means in physical terms, consider the scale. A single liter of Gulf water, held in a container, would contain more Karenia brevis organisms than the entire human population of the Western Hemisphere. Each cell measures between 20 and 40 micrometers in diameter, invisible to the naked eye individually, but collectively capable of altering the chemistry, color, and oxygen content of thousands of square kilometers of open water. The spatial extent of the bloom was confirmed through multiple independent data streams converging at the same time. NOAA's Coastwatch program, which processes satellite-derived ocean color data from instruments aboard NASA and ESA platforms, had been tracking elevated chlorophyll A concentrations in the eastern Gulf of Mexico since mid-July. Chlorophyll A is the primary photosynthetic pigment shared by nearly all marine algae. When its concentration rises sharply across a defined geographic area, it signals rapid algal multiplication. Satellite passes from the MODIS Aqua and Terra instruments, operating at a spatial resolution of approximately 250 to 500 meters per pixel, showed a coherent mass of elevated biomass extending from the waters off Charlotte County southward through Lee County and approaching the outer reaches of Collier County. By early August, that surface expression covered an estimated area of several hundred square kilometers. It was visible from orbit as a distinct, irregular patch of altered ocean color, positioned between the shoreline and waters approximately 20 to 30 kilometers or 12 to 19 miles offshore. Satellite data alone cannot identify a species. It measures light reflectance and chlorophyll concentration, not taxonomy. The species confirmation came from water samples collected by research vessels operated by Mote Marine Laboratory in Sarasota and by Florida Gulf Coast University monitoring teams in Fort Myers. These samples were analyzed using light microscopy and, in some cases, flow cytometry, a technique that passes individual cells through a laser beam and identifies them by their optical signature. The identification was unambiguous. Karenia brevis dominated the samples across multiple collection sites. In some nearshore locations, the organism accounted for more than 90% of the total phytoplankton cell count. That degree of dominance is significant. A healthy phytoplankton community contains dozens of species competing for light and nutrients. When a single species reaches this level of concentration, it indicates that the environmental conditions have tilted decisively in its favor. Those conditions were measurable and interlocking. Sea surface temperatures in the bloom zone remained between 29 and 31 degrees Celsius, or approximately 84 to 88 degrees Fahrenheit, throughout early August. Wind speeds were low. The Gulf surface was relatively calm, which reduced vertical mixing and allowed the bloom to stratify into a dense layer in the upper 2 to 5 meters of the water column. Calm conditions also meant reduced wave energy, which limited the physical disruption that might otherwise break the bloom apart. Karenia brevis is classified as a mixotroph. It can photosynthesize using sunlight, but it can also take up dissolved organic compounds from the surrounding water. This dual feeding strategy gives it a competitive advantage under nutrient conditions that would slow purely autotrophic algae. The nutrient-enriched nearshore waters, still receiving elevated nitrogen and phosphorus inputs from the Caloosahatchee River system, provided a replenishing supply of dissolved material. The bloom was not depleting its resource base. It was being sustained. Water sampling data collected at monitoring stations near Sanibel Island and Cape Coral during the first 2 weeks of August documented another measurable shift. pH levels in the bloom zone were rising during daylight hours, sometimes reaching values above 8.5. That rise is a direct product of photosynthetic activity. As Karenia brevis cells fix carbon dioxide from the surrounding water, they alter the local carbonate chemistry. At the same time, nighttime respiration by the dense algal mass consumed dissolved oxygen, pushing concentrations below 3 mg per liter in some bottom water zones. The daily oscillation between these two chemical states placed significant physiological stress on fish and invertebrates unable to relocate. The bloom's behavior was not uniform across its area. Satellite passes captured visible internal structure. The densest concentrations appeared as elongated filaments and irregular patches, shaped by small-scale circulation features, tidal flow through inlets, and the geometry of the coastline itself. Charlotte Harbor, a large shallow estuary covering approximately 700 square kilometers, or 270 square miles, was acting as a partial trap. Water entering the harbor on incoming tides carried bloom cells inward. Outgoing tides returned them to the nearshore zone. The estuary was cycling the bloom through its interior repeatedly. This recirculation pattern mattered for what the bloom was producing. Karenia brevis cells generate brevetoxins continuously as a byproduct of normal cellular metabolism. As cell density increased through early August, the total toxin load dissolved in the water column was rising in proportion. Toxin measurements from water samples collected by Mote Marine Laboratory showed brevetoxin concentrations in the range of tens to hundreds of nanograms per liter in the densest bloom zones. That range falls within the region associated with physiological effects in sensitive marine species. At this stage, in early August 2018, the bloom was confirmed, mapped, and chemically characterized. State agencies issued the first formal harmful algal bloom advisories for Lee and Charlotte counties. The advisories recommended avoiding contact with discolored water and warned that shellfish harvesting in the affected areas was under review. The advisory language was measured and procedural, but the data behind it described a bloom that was not weakening. It was large, it was dense, it was chemically active, and the physical conditions sustaining it had not changed. The next question was not whether the bloom would affect the coastline more severely. The question was how quickly and across how much of it. A Karenia brevis cell does not deploy its toxin as a defense mechanism in the way a jellyfish deploys venom. The production of brevetoxins is not a targeted biological response. It is metabolic, a continuous chemical output generated as part of the organism's normal cellular processes, regardless of whether a predator is present, whether conditions are favorable, or whether the surrounding water is open ocean or an enclosed estuary. The toxin is always being made. What changes is the scale at which it accumulates. Brevetoxins belong to a class of organic compounds called polyether ladder toxins. Their molecular architecture is unusually complex. Each molecule consists of a rigid, fused ring structure containing between 10 and 11 ether-linked rings arranged in a linear chain. This configuration is not accidental. It is precisely shaped to interact with a specific target in animal physiology. And that specificity is what makes brevetoxins effective at extraordinarily low concentrations. The target is the voltage-gated sodium channel. These channels are protein structures embedded in the cell membranes of nerve and muscle tissue across virtually all vertebrate animals. Under normal conditions, sodium channels open briefly in response to an electrical signal, allow a rapid influx of sodium ions into the cell, and then close. That opening and closing cycle is the fundamental mechanism behind nerve impulse transmission and muscle contraction. It is how a fish moves its fins, how a dolphin regulates its breathing, and how a sea turtle coordinates its dive. Brevetoxin binds to a specific site on the sodium channel, designated Site 5 on the channel's alpha subunit. Once bound, it holds the channel in a persistently open state. Sodium ions continue flowing into the cell without the normal regulatory closure. The result is continuous, uncontrolled depolarization of the cell membrane. Nerve cells fire repeatedly without pause. Muscle fibers contract without release. The signal that should be a brief, controlled pulse becomes a sustained, unregulated cascade. In fish, this mechanism produces a recognizable and well-documented sequence of physiological failure. Early exposure causes erratic swimming behavior, loss of coordination, circular motion, surface orientation, where the animal struggles to maintain depth. This is sometimes described in field observations as spinning disease, though it is not a disease in the infectious sense. It is a direct neurological consequence of channel dysfunction. As exposure continues and toxin concentration in tissue rises, the fish loses the ability to maintain buoyancy. Respiratory muscles, including those controlling gill movement, begin to fail. Death follows from respiratory arrest. The concentration at which this sequence begins in fish is measurably low. Laboratory studies have documented lethal effects in sensitive species at brevetoxin concentrations of approximately 1 microgram per liter of water. In the bloom zones mapped around Charlotte Harbor and the Lee County nearshore waters during early to mid-August 2018, dissolved brevetoxin concentrations measured from water samples were in the range of 10 to several hundred nanograms per liter, approaching and in some locations exceeding biologically relevant thresholds for fish exposure. Those measurements capture only part of the toxin picture. Brevetoxins exist in the water column in multiple forms simultaneously. Dissolved toxins are present as free molecules in solution. Particulate toxins are bound within living and dead Karenia brevis cells. And a third fraction accumulates in the tissues of organisms that have filtered or ingested bloom-associated material. Each fraction behaves differently in the environment, moves at different rates, and presents different risks to different parts of the food web. Filter feeders are particularly efficient at concentrating the particulate fraction. Oysters, clams, and mussels process large volumes of water continuously. A single adult oyster can filter up to 190 liters, or approximately 50 gallons of water per day. As it filters, it retains Karenia brevis cells and the toxins bound within them. The brevetoxins accumulate in the oyster's tissue at concentrations that can exceed those in the surrounding water by a factor of several thousand. This process, called bioconcentration, does not harm the bivalve itself. Shellfish lack the voltage-gated sodium channel configuration found in vertebrates and are largely insensitive to brevetoxin at environmentally relevant doses. But the toxin stored in their tissue is fully active when ingested by an animal that is sensitive to it. By the second week of August 2018, Florida's Division of Aquaculture and the Department of Agriculture and Consumer Services had placed shellfish harvesting closures on waters in Lee, Charlotte, and Collier counties. The closures were triggered by regulatory thresholds, specifically tissue concentrations of brevetoxin exceeding 20 mouse units per 100 grams of shellfish flesh, the established safety limit under the National Shellfish Sanitation Program. In some sampled oyster beds near Pine Island Sound, concentrations were reported above that threshold. The toxin's behavior in open water introduced an additional complication for marine monitoring teams. Brevetoxins are not rapidly degraded in the marine environment. Under typical Gulf of Mexico conditions in summer, dissolved brevetoxin molecules remain chemically stable for days to weeks. They adsorb onto particulate organic matter and suspended sediment, which can redistribute them through tidal action and bottom currents independently of the living bloom. This means toxin presence in the water column can persist and can precede and follow the peak bloom density at any given location. Marine mammal researchers monitoring the area during this period noted behavioral changes in bottlenose dolphin groups in and around Charlotte Harbor. Dolphins, as air-breathing apex predators, are exposed to brevetoxins through two pathways simultaneously. They ingest toxins by consuming affected fish, and they inhale aerosolized toxin particles at the water surface when they breathe. Their neurological sensitivity to brevetoxin is well established from prior red tide events in Florida waters. In 2013 and 2014 alone, brevetoxin exposure was implicated in the deaths of more than 100 bottlenose dolphins along Florida's Gulf Coast. In early August 2018, Dolphin strandings had not yet reached critical numbers, but the toxin loading in the system was rising. The bloom was delivering brevetoxins into the food web from the base upward, through plankton, through filter feeders, through forage fish, toward the larger predators at the top. The chemical signal was moving through the ecosystem faster than its biological consequences were yet visible on the surface. There is a structural feature of polyether ladder toxins that marine toxicologists consider particularly significant. Unlike many marine biotoxins that are excreted relatively quickly, brevetoxins and their metabolic derivatives can accumulate in vertebrate tissue across repeated sublethal exposures. An animal does not need to encounter a single lethal dose. Sequential exposures, each insufficient to cause immediate death, can build toward physiological failure over days. The bloom did not need to be uniformly dense to be lethal. It needed only to be present, persistent, and widely distributed across the feeding and breathing range of the animals living within it. By mid-August, those conditions had been met across a coastline stretching more than 100 kilometers, or approximately 62 miles. The toxin was in the water, in the sediment, in the shellfish tissue, and in the bodies of fish moving through the bloom zone. What had been chemically diffuse in July was now concentrated, widespread, and biologically active throughout the water column. The system had loaded. The consequences of that loading were about to become visible at a scale that water sample data and laboratory measurements had not yet fully communicated to the public watching from shore. Until mid-August 2018, the crisis unfolding in the waters off Florida's Gulf Coast was primarily a subsurface event. The toxins were in the water column, in shellfish tissue, in the bodies of fish. The people standing on the beach could see the discolored water and the dead fish accumulating along the tide line. What they could not yet perceive was that the bloom had found a way to cross the boundary between ocean and atmosphere, and that the air itself was becoming a delivery mechanism. The process responsible for this transition is called aerosolization. It is not unique to red tide events. Breaking waves routinely inject microscopic droplets of seawater into the lower atmosphere. These droplets, called sea spray aerosols, form when a wave crest collapses and the trapped air bubble bursts at the surface. Each bursting bubble ejects a jet of water that fragments into thousands of droplets, ranging in diameter from less than 1 micrometer to several hundred micrometers. The smallest droplets evaporate almost instantly, leaving behind a residue of whatever was dissolved or suspended in the original water. In bloom conditions, that residue includes brevetoxin fragments. As Karenia brevis cells concentrate at the ocean surface and are disrupted by wave action, their cellular contents are released directly into the spray generation zone. Brevetoxin molecules, along with cell membrane fragments carrying bound toxins, become incorporated into sea spray droplets. Droplets smaller than approximately 10 micrometers in aerodynamic diameter do not settle quickly. They remain suspended in the lower atmosphere and travel with onshore winds. Wind direction and speed during the second and third weeks of August 2018 were critical factors. Meteorological data from National Weather Service stations along the southwest Florida coast recorded consistent onshore flow from the west and southwest, with average wind speeds between 10 and 20 kilometers per hour, or approximately 6 to 12 miles per hour. That wind regime was sufficient to carry aerosol-sized particles from the surf zone inland by several hundred meters to more than a kilometer under sustained conditions. Beaches facing directly into the prevailing onshore flow received the highest aerosol concentrations. Fort Myers Beach, Bonita Beach, and the beaches of Sanibel Island were among the first locations where public health effects became formally documented. By the third week of August, the Lee County Department of Health had begun receiving a measurable increase in calls and clinic visits reporting respiratory symptoms. The pattern was consistent across reports. Individuals spending time on or near the beach developed coughing, sneezing, and a burning or irritating sensation in the throat and upper airway within minutes of exposure. In many cases, symptoms resolved within 30 to 60 minutes of moving indoors or away from the shoreline. That symptom profile and its reversibility are characteristic of brevetoxin aerosol exposure. Brevetoxin acts on the respiratory epithelium through the same sodium channel mechanism that affects fish nerve tissue, though in humans the exposure route and concentration determine the severity of the response. At the aerosol concentrations typical of a moderate red tide event, effects are generally limited to the upper respiratory tract. The toxin stimulates sensory nerve endings in the nasal passages, throat, and bronchi, triggering reflexive coughing and sneezing. The sensation is often described by those who experience it as similar to inhaling finely ground pepper or a chemical irritant. For individuals with pre-existing respiratory conditions, the response was more severe. Asthma is the condition most extensively documented in clinical literature on brevetoxin aerosol exposure. Research conducted following previous Florida red tide events had demonstrated that brevetoxin inhalation causes measurable bronchoconstriction in asthmatic subjects at concentrations that produce only mild irritation in healthy individuals. The mechanism is direct. Sodium channel activation in bronchial smooth muscle causes constriction of the airway. In an asthmatic patient whose airways are already hyperreactive, this additional stimulus can trigger significant bronchospasm. By late August 2018, emergency department data from Lee Memorial Hospital and Cape Coral Hospital showed elevated respiratory complaint volumes compared to the same period in prior years. Health advisories issued by the Florida Department of Health recommended that individuals with asthma, chronic obstructive pulmonary disease, or other respiratory conditions avoid coastal beaches entirely during active red tide conditions. Those advisories carried weight. They were not precautionary language applied to a theoretical risk. They were responses to a documented and ongoing pattern of hospital presentations. Quantifying the actual airborne brevetoxin concentration at any given beach location during this period was technically difficult. Air sampling for brevetoxins requires specialized collection equipment, typically a high-volume air sampler fitted with a glass fiber filter, followed by solvent extraction and analysis by liquid chromatography with mass spectrometry detection. Mote Marine Laboratory maintained an air monitoring program that had been operational since earlier red tide events, and sampling data from stations near Sarasota and the southern Lee County coast confirmed detectable brevetoxin concentrations in air samples collected during onshore wind events. Measured concentrations at beach locations during active surf conditions ranged from the low nanogram to low microgram per cubic meter range, sufficient to produce effects in sensitive individuals within minutes of continuous exposure. The spatial extent of the aerosol hazard was governed by the same oceanographic factors controlling the bloom itself. Sections of coastline with high wave energy, direct westward exposure, and proximity to the densest bloom concentrations received the highest aerosol loading. Protected areas behind barrier islands, or stretches of coast sheltered from the prevailing wind by geography, showed lower exposure. The hazard was not uniform. It was patchy, variable with wind shifts, and capable of changing rapidly as atmospheric conditions evolved through the day. Thermal stratification of the lower atmosphere added another layer of complexity. During the day, solar heating of the beach surface created a shallow, convective boundary layer that mixed aerosols vertically and diluted their concentration. In the early morning hours before solar heating began, the atmosphere near the surface was more stable. Aerosols accumulated in a shallow layer close to the ground. Several health reports from this period describe particularly acute symptoms in early morning beach walkers, consistent with that stratification pattern. The social consequences of the aerosol hazard were immediate and visible in ways that the subsurface toxin loading had not been. Water discoloration and dead fish had been reported by those who came to the shore to observe them. Respiratory irritation was experienced by people who had no particular interest in the bloom and no prior awareness of its severity. Tourists who had traveled to Southwest Florida for a beach holiday encountered symptoms within minutes of stepping onto the sand. Local residents who walked their dogs along the tide line in the morning returned home coughing. The beach, for coastal communities whose economic and social identity is inseparable from direct access to the Gulf, had become a place that made people sick. That shift in the lived experience of the coastline produced a public response that data releases and environmental advisories had not. People stopped going to the beach. And those who ran businesses dependent on that foot traffic began to register with increasing urgency that something had gone fundamentally wrong with the summer of 2018. Offshore, the bloom continued to expand. Its western and southern margins were advancing along the coast toward Collier County and Naples. Its northern extent was approaching Sarasota County waters. The surface area of elevated Karenia brevis concentration, as measured in the most recent satellite passes processed by NOAA's Coastal Monitoring Program, had grown to an estimated area exceeding 1,000 square kilometers, or approximately 390 square miles. The toxin was now traveling by air and by water simultaneously, and the coastline that had first registered isolated fish deaths 6 weeks earlier was now contending with a hazard that extended from the seafloor to the atmosphere above the beach. In the final week of August 2018, the accumulation of conditions that had been building across 6 weeks of bloom growth reached a biological threshold. The toxin loading documented in the water column, the shellfish tissue, and the bodies of forage fish had moved far enough up the food web and had persisted long enough in the environment that the mortality events it was generating could no longer be described as isolated. What began appearing along the shorelines of Lee, Charlotte, Sarasota, and Collier counties in late August and through September was systematic, it was species-wide, and it was occurring simultaneously across more than 150 kilometers, or approximately 93 miles, of coastline. The fish mortality came first in visible scale. Snook, redfish, tarpon, sheepshead, grouper, and Goliath grouper, species occupying different depth zones, different habitat types, and different positions in the food web, were dying in numbers that field observers from the Florida Fish and Wildlife Conservation Commission described as unlike anything documented in the region's recent monitoring history. A single aerial survey conducted over Lee County waters in late August counted thousands of individual fish carcasses visible from low altitude across a stretch of coastline less than 20 kilometers or 12 miles long. That count represented only what was visible at the surface at the moment of the overflight. It did not include animals that had sunk, animals dying in deeper water offshore, or animals already removed by scavengers. Goliath grouper, a species that can exceed 180 kilograms, or nearly 400 pounds, and live for more than 35 years, were among the most visually striking casualties. These animals are not easy to kill. They are physiologically robust, habitat-specific, and largely sedentary as adults, anchored to reef and hard-bottom structures in relatively shallow nearshore water. Their mortality in significant numbers indicated that the brevetoxin concentration in the water column had reached levels sufficient to affect even large, long-lived animals through sustained passive exposure across their entire home range. They could not relocate. The bloom had moved to them. The mechanism of death in fish exposed to elevated dissolved brevetoxin concentrations had been established in controlled laboratory studies. At water concentrations above approximately 10 micrograms per liter, complete respiratory failure in most marine fish species occurs within hours. Measurements from the densest bloom zones in Lee County waters during late August were approaching and in some locations exceeding that threshold. Field observations of dying fish showed the characteristic neurological progression described in earlier chapters: loss of equilibrium, surface rolling, cessation of coordinated movement, then stillness. The accumulation of carcasses along the tide line introduced a secondary biological process that compounded the oxygen depletion already underway in the water column. Decomposing organic matter consumes dissolved oxygen as aerobic bacteria break down tissue. Each large fish carcass represents a localized oxygen sink. Along stretches of beach where thousands of carcasses were concentrated by tidal action and longshore drift, the decomposition load was sufficient to drive dissolved oxygen in adjacent shallow water to near zero levels. Those hypoxic conditions killed additional organisms that had survived the direct toxin exposure, including invertebrates, juvenile fish sheltering in the shallows, and bottom-dwelling species unable to move to oxygenated water. The smell produced by this scale of decomposition was documented in health complaint records filed with county agencies and in local news reporting throughout September 2018. Hydrogen sulfide and ammonia, the primary gaseous byproducts of protein decomposition in warm water, were detectable at distances of several hundred meters from the most heavily affected beaches. At Fort Myers Beach, Bonita Beach, and sections of Sanibel Island, the combination of brevetoxin aerosol irritation and decomposition gases created air quality conditions that health officials classified as a compound respiratory hazard. These were not independent problems. They were simultaneous outputs of the same biological event. The marine mammal mortality that emerged through August and September carried a different kind of significance. Bottlenose dolphins began stranding along the southwest Florida coast in numbers that triggered an unusual mortality event investigation by NOAA's National Marine Fisheries Service. An unusual mortality event, or UME, is a formal federal designation applied when marine mammal strandings are unexpected, involve a significant die-off, and require an immediate response. The designation activates a coordinated federal and state response network and mandates systematic necropsy of stranded animals. Dolphin stranding during this period showed a consistent pattern in post-mortem analysis. Tissue samples from multiple animals revealed brevetoxin concentrations in liver, lung, and blubber tissue at levels consistent with lethal or near-lethal exposure. Lung pathology in several animals showed hemorrhagic lesions and inflammatory changes characteristic of inhalation toxicity, the consequence of repeatedly breathing at the surface of water with high aerosol toxin loading. The dual exposure pathway that had been flagged as a concern in earlier monitoring was confirmed in necropsy findings. These animals had been poisoned both by what they ate and by the air above the water where they breathed. Sea turtle strandings followed a trajectory that marine biologists found particularly alarming when the numbers were compiled through August and September. Loggerhead, Kemp's Ridley, and green sea turtles were found stranded or floating debilitated across the coastline. Sea turtles are exposed to brevetoxin primarily through ingestion, consuming fish, jellyfish, and invertebrates carrying accumulated toxin loads. Their neurological response to brevetoxin follows the same sodium channel mechanism documented in other vertebrates, producing loss of coordination, inability to dive, and eventual respiratory failure in animals unable to reach the surface to breathe. The Florida Fish and Wildlife Conservation Commission and partner organizations, including Mote Marine Laboratory, the Clinic for the Rehabilitation of Wildlife on Sanibel, and Sea Turtle Conservancy recovery teams, were collecting and transporting live but debilitated turtles to rehabilitation facilities. Some animals recovered with supportive care once removed from the exposure environment. Others did not. Total sea turtle strandings attributable to the 2018 red tide event across southwest Florida ultimately numbered in the hundreds across the full duration of the bloom. Manatee mortality during this period added a species of particular conservation concern to the casualty record. Florida manatees, listed as a threatened species under the Endangered Species Act, with a total estimated population of fewer than 8,000 individuals at the time, were dying in bloom-affected waters through August and September. Manatees are exposed to brevetoxin primarily through ingestion of seagrass and other submerged aquatic vegetation on which toxin-containing particles had settled. Their large body mass and slow metabolic rate do not confer protection from brevetoxin. Necropsy results from stranded manatees confirmed brevetoxin in multiple tissue compartments. At the population level, each confirmed brevetoxin-related manatee death represented a measurable impact on a species with limited reproductive capacity, a slow reproducing animal losing individuals faster than natural recruitment could offset. The physical condition of the shoreline through late August and September 2018 was documented extensively in local government records, wildlife agency reports, and journalistic accounts. Beaches that had been maintained as recreational amenities through the early summer were now covered in biological debris at a scale that exceeded the capacity of routine municipal cleanup operations. Lee County, responding to the scope of the accumulation, deployed heavy equipment, front-end loaders, and dump trucks to remove fish carcasses from the most heavily affected public beaches. In a single week in early September, county operations removed an estimated 174 metric tons, or approximately 383,000 pounds, of dead marine life from Lee County beaches alone. That figure did not include material removed from private shorelines, from water surfaces, or from areas outside the county's operational reach. The removal operation itself created logistical and environmental complications. Decomposing fish carcasses could not simply be taken to a standard landfill without risk of leachate contamination and regulatory violation. The material required processing as organic waste, with some volume directed to composting facilities and some to permitted disposal sites. The volume exceeded the immediate capacity of available facilities, creating backlogs that extended the period during which decomposing material remained on the beach or in temporary staging areas. For the ecosystems themselves, the die-off was not only a mortality event, it was a structural disruption. Snook and redfish populations in Charlotte Harbor and the surrounding estuaries had taken years to recover from previous red tide impacts and from fishing pressure regulated to allow that recovery. The concentrated loss of adult fish across multiple species in a single season removed animals that represented years of growth and reproductive potential. Seagrass beds, already stressed by turbidity created by the bloom reducing light penetration, were losing the grazing and disturbance dynamics that fish communities provide. Reef and hard-bottom habitats that had supported Goliath grouper populations were now vacant. The bloom had not merely passed through the ecosystem, it had removed significant components of it, and as September continued, the mortality was not declining. The Karenia brevis cell counts in the water column remained above 1 million cells per liter across large portions of the affected zone. The bloom was holding. The conditions sustaining it had not yet shifted, and the coastline, stripped of much of its visible marine life and covered in the evidence of what the water had done, was about to register the full weight of the event in the economy and public health of the communities that depended on it. The marine food web is not a simple linear chain. It is a network of overlapping dependencies where energy and chemical compounds move upward through predation, laterally through scavenging and filter feeding, and downward through the settling of organic particles to the sea floor. When a persistent, harmful algal bloom saturates that network with a stable, bioaccumulative neurotoxin for weeks, the contamination does not follow a single pathway. It follows all of them simultaneously. By early September 2018, brevetoxin had been confirmed in the tissues of organisms representing every functional feeding group in the Southwest Florida coastal ecosystem. Filter feeders had concentrated it from the water column. Herbivores had ingested it through contaminated vegetation. Predatory fish had accumulated it through repeated consumption of affected prey. Scavengers feeding on bloom-killed carcasses were receiving concentrated doses from already toxic tissue. The toxin had not entered the food web at a single point. It had permeated the system from multiple entry points and was now present at every level. The filter feeder situation required the most immediate regulatory response. Eastern oysters in Pine Island Sound, Estero Bay, and Charlotte Harbor had been accumulating brevetoxin since the bloom first reached nearshore concentrations in late July. By September, tissue samples analyzed under the National Shellfish Sanitation Program showed concentrations substantially exceeding the closure threshold of 20 mouse units per 100 grams of shellfish flesh. In some beds near the Caloosahatchee River mouth, concentrations were reported at multiples of that threshold. The Florida Department of Agriculture and Consumer Services responded with emergency shellfish harvesting closures covering waters in Lee, Charlotte, Collier, and Sarasota counties. These closures applied to all bivalve shellfish species, oysters, clams, and bay scallops. Commercial shellfish operations in the affected counties ceased entirely. Leaseholders who had invested in gear, seed stock, and growing infrastructure found their inventory contaminated and unsellable. The closures would remain in effect until tissue sampling confirmed brevetoxin concentrations had returned below regulatory thresholds, a process that required not only the bloom to decline, but sufficient time for shellfish to metabolically clear accumulated toxins. Studies of brevetoxin clearance indicate that tissue concentrations can remain above regulatory thresholds for weeks to months after the bloom subsides. The contamination was not a fixed deposit. It was being continuously replenished. Commercial fishing closures extended beyond shellfish. Florida's Fish and Wildlife Conservation Commission implemented emergency advisories covering finfish harvesting in bloom-affected waters. For commercial fishermen operating out of Fort Myers, Cape Coral, and smaller communities along Lee and Charlotte counties, the practical effect was a near-total suspension of economically viable activity. Processing facilities temporarily halted operations due to lack of supply. The boats stayed at the dock. The bioaccumulation dynamics operating within the food web were not limited to direct toxin transfer through predation. Brevetoxin molecules released through decomposition of bloom-killed fish re-entered the dissolved and particulate pools in the water column, available for reuptake by filter feeders and readsorption onto sediment particles. Water quality sampling by Florida Gulf Coast University researchers detected brevetoxin in sediment cores from Charlotte Harbor and Pine Island Sound. Sediment-bound toxin can be resuspended into the water column by tidal current, storm disturbance, or bioturbation from bottom-feeding organisms, creating a toxin reservoir that persists after the living bloom has declined. The bloom's chemical legacy extended its ecological impact by weeks to months beyond its visible duration. Seagrass beds in the bloom zone were experiencing compound stress beyond toxin contamination. Karenia brevis at high cell densities significantly reduces light penetration through the water column, creating an effective shading layer over submerged vegetation. Seagrasses depleted of sufficient light lose root energy reserves, causing leaf dieback and, in severe cases, permanent bed loss. Post-bloom surveys documented measurable declines in seagrass cover in portions of Pine Island Sound and Estero Bay. Those losses mattered beyond the vegetation itself. Seagrass beds serve as nursery habitat for juvenile snook, redfish, and grouper. The zooplankton community, which forms the critical intermediate layer between microscopic phytoplankton and juvenile fish, showed significant disruption in plankton samples analyzed by Mote Marine Laboratory. Copepods and small crustaceans that normally graze on phytoplankton are sensitive to brevetoxin and are killed or repelled by dense bloom water. Their depletion removes a fundamental energy transfer step in the food web. Juvenile fish, depending on zooplankton as a primary food source, face starvation pressure independently of any direct toxin exposure. The functional food web of Southwest Florida's coastal waters had not simply been reduced in biomass. It had been disrupted at every level simultaneously. Filter feeders contaminated, forage fish eliminated, zooplankton depleted, seagrass stressed, apex predators poisoned, and sediment chemically marked. Recovery would not be a matter of waiting for the bloom to clear. The structural damage ran deeper than any single season could repair. By September 2018, the consequences of the bloom had moved decisively onshore. The marine ecosystem had been absorbing the biological damage for weeks. Now, the human systems built along that coastline, the hospitals, the fishing businesses, the hotels, the restaurants, the municipal governments, were registering the same event through a different set of measurements. The public health effects were the most immediate and the most direct. Lee County Department of Health data from August and September showed emergency department visits for respiratory complaints running significantly above the 5-year average for the same period. The presenting symptoms were consistent across cases: coughing, throat irritation, shortness of breath, and eye irritation in individuals who had spent time near the beach. In most healthy adults, symptoms resolved within hours of leaving the coastal environment. In patients with asthma or chronic obstructive pulmonary disease, the presentations were more acute. Several required bronchodilator treatment before discharge. A smaller number required observation beyond the initial emergency department visit. Lee Memorial Hospital and Gulf Coast Medical Center in Fort Myers both reported elevated respiratory intake volumes through the peak bloom weeks. The Florida Department of Health issued successive public health advisories recommending that sensitive populations avoid coastal areas entirely. Those advisories covered a coastline that, in normal circumstances, those same populations had been encouraged to visit for its therapeutic and recreational value. The reversal was not lost on the communities receiving it. Beyond respiratory effects, the bloom generated a secondary public health concern through fish consumption advisories. The Florida Department of Health issued guidance stating that fish caught in bloom-affected waters should not be consumed, and that any fish showing neurological symptoms should not be handled without gloves. Calls to the Florida Poison Control Center increased during this period, with reports of individuals who had consumed shellfish or fish from affected areas and presented with symptoms consistent with neurotoxic shellfish poisoning, including tingling and numbness of the lips and extremities, dizziness, and gastrointestinal distress. Neurotoxic shellfish poisoning caused by brevetoxin ingestion is rarely fatal in otherwise healthy adults, but it is clinically significant and its symptoms can persist for days. The economic damage accumulated through a different kind of arithmetic. Southwest Florida's coastal economy is structured around seasonal tourism, with late summer and fall representing a transitional period between the peak winter season and the slower summer months. The 2018 bloom arrived at precisely the moment when the region would normally be preparing for the return of its most economically significant visitor demographic. Hotel occupancy data from Lee County Tourist Development Council records showed significant year-over-year declines through August and September. Properties on Fort Myers Beach and Sanibel Island, positioned directly on the most heavily affected shoreline, reported cancellation rates that operators described as without precedent in recent memory. Vacation rental platforms showed a pattern of last-minute cancellations from guests who had monitored news coverage of the bloom and chosen not to travel. The beaches that were the primary reason those guests had booked their accommodations were covered in dead fish and generating air that made people cough. Restaurant and retail businesses in beach communities reported revenue declines ranging from 30 to more than 60% compared to the same period in prior years, according to accounts collected by the Lee County and Collier County Chambers of Commerce. Establishments that depended on walk-in traffic from beach visitors had no substitute customer base to draw on. Several temporary closures were reported among smaller operators who could not sustain fixed operating costs against that revenue drop. The commercial fishing industry, whose closures had been detailed in the previous chapter's regulatory context, translated those closures directly into household income loss. Charter fishing operations, which generate significant revenue during fall months when offshore fishing conditions are typically favorable, were unable to take clients out on water that was visibly discolored and producing dead fish. Some operators reported complete booking voids for September and into October. For captains and deckhands whose annual income is concentrated in a few peak months, a complete loss of September revenue represented a structural financial injury, not merely a temporary inconvenience. Lee and Collier counties submitted formal requests to the state of Florida for emergency economic assistance declarations. Governor Rick Scott issued an emergency declaration for 7 affected counties in August, which activated state resources for cleanup operations and opened pathways for federal assistance applications. The total economic impact of the 2018 red tide event on Southwest Florida was subsequently estimated by state and academic analysts at over $1 billion, when direct losses across tourism, fishing, aquaculture, and associated service industries were aggregated. What the economic data could not fully capture was the cumulative toll on communities whose identity, livelihood, and daily life are inseparable from the water that had now turned against them. For the people who had built their lives on the Gulf Coast, the summer of 2018 had demonstrated, with unusual clarity, how completely a biological event occurring in the water column could dismantle the human systems arranged along its edge. The scientific infrastructure monitoring Florida's coastal waters did not mobilize in response to the 2018 bloom. It had been running continuously before the bloom began, and it was the data produced by that infrastructure that first confirmed what fishermen and residents had started reporting in July. What changed through August and September was the intensity of that monitoring effort, the number of institutions contributing to it, and the degree to which real-time data was being used to drive regulatory and public health decisions on a day-to-day basis. Mote Marine Laboratory in Sarasota operates one of the longest-running harmful algal bloom monitoring programs on the Gulf Coast. Its network of water sampling stations, maintained by research staff and trained volunteer observers, had been collecting weekly Karenia brevis cell counts from Southwest Florida nearshore waters for years before 2018. When cell counts began rising in late July, that baseline data provided the critical context for interpreting the rate of increase. Without a multi-year record of background concentrations, distinguishing an emerging bloom from normal seasonal variation would have taken significantly longer. By August, sampling frequency at priority stations had increased from weekly to daily at several locations. Research vessels from Mote, Florida Gulf Coast University, and the Florida Fish and Wildlife Research Institute were conducting coordinated transect surveys, systematic sampling runs along defined lines extending from the shoreline to several kilometers offshore. Each transect collected water samples at multiple depths, measured temperature and salinity profiles, and recorded dissolved oxygen. The resulting dataset allowed researchers to map the three-dimensional structure of the bloom across the water column, not merely its surface expression. NOAA's Harmful Algal Bloom Operational Forecast System, known as HABSOS, integrated that field sampling data with satellite-derived ocean color imagery and numerical ocean circulation models to produce bloom forecasts updated several times per week. Those forecasts estimated the probable movement of the bloom over the following 72 to 96 hours, based on predicted wind patterns, surface current trajectories, and the observed position of the bloom's leading edge. The forecasts were not precise at the scale of individual beaches, but they provided coastal managers, health departments, and fishing regulators with advance indication of which areas were likely to experience elevated cell counts in the near term, allowing some degree of anticipatory advisory issuance rather than purely reactive response. The toxin monitoring program running parallel to cell count surveillance required a different set of analytical methods. Brevetoxin quantification in water samples, shellfish tissue, and air filters required liquid chromatography coupled with tandem mass spectrometry, an analytical technique capable of detecting and quantifying specific toxin congeners at concentrations in the nanogram per liter range. The Florida Department of Agriculture and Consumer Services Laboratory in Tallahassee processed shellfish tissue samples submitted from closure zones on a schedule that, during the peak of the event, involved dozens of samples per week. Turnaround time between sample collection and regulatory decision was a critical operational variable. Delays in laboratory processing directly extended the period of uncertainty for aquaculture operators and shellfish harvesters awaiting reopening determinations. Air quality monitoring for aerosolized brevetoxins presented particular technical challenges. Unlike water sampling, which could be conducted from a vessel or a fixed pier, meaningful air sampling required equipment positioned at beach locations during active onshore wind events, conditions that were logistically demanding and that produced results reflecting highly variable exposure concentrations, depending on wind speed, wave height, and proximity to the surf zone. Mote Laboratories' air monitoring stations collected filter samples that, when analyzed, confirmed detectable brevetoxin concentrations at beach locations during peak bloom conditions. Academic researchers from the University of South Florida, Florida Atlantic University, and the University of Miami contributed additional monitoring capacity through their own vessel-based sampling programs and through analysis of archived satellite data. Remote sensing researchers examined sequences of MODIS and Sentinel satellite imagery to reconstruct the spatial development of the bloom from its offshore origins in late 2017 through its coastal impact in 2018. That retrospective analysis identified the approximate timing and pathway of onshore transport, contributing to post-event understanding of how the bloom's trajectory might have been anticipated earlier. The collective monitoring effort of the 2018 event generated a dataset of unusual density and duration for a single bloom episode. It documented, in quantitative terms, how a harmful algal bloom moves through a coastal system, loads toxins into a food web, and generates measurable effects at the human interface. That documentation served both the immediate management response and the longer-term scientific record. What the data also revealed, without ambiguity, was that the tools available for tracking the bloom were substantially more developed than the tools available for stopping it. Monitoring had become precise. Intervention remained beyond reach. Through the first 3 weeks of September 2018, the environmental conditions sustaining the bloom had remained essentially stable. Water temperatures were still elevated. Onshore wind patterns were still delivering bloom-laden water to the coastline. Nutrient inputs from freshwater discharges were still supplementing the nearshore chemical environment. A bloom does not exhaust itself simply by causing damage. It persists as long as the physical and chemical conditions supporting its growth remain favorable. What ended the 2018 bloom was not a single event. It was a convergence of shifting environmental variables that together removed the conditions the bloom required to sustain itself. The first measurable signal came from sea surface temperature data recorded by NOAA buoys and satellite thermal infrared sensors in late September. Water temperatures in the bloom zone, which had remained between 29 and 31 degrees Celsius, or 84 to 88 degrees Fahrenheit, through August and early September, began a gradual decline. By the final week of September, surface temperatures at several nearshore stations had dropped to approximately 27 degrees Celsius, or 80 degrees Fahrenheit. That reduction sounds modest. In the context of phytoplankton physiology, it is not. Karenia brevis grows most efficiently within a relatively narrow thermal window. The late September temperatures were still well above its lower growth threshold, but the directional trend had reversed. The thermal advantage supporting rapid cell division through the summer was weakening. The atmospheric pattern shift accompanying the temperature decline was more immediately significant for bloom transport dynamics. Through August and much of September, the dominant wind regime had been from the west and southwest, onshore flow continuously delivering bloom-concentrated surface water toward the coastline. In late September, a series of frontal passages associated with early autumn atmospheric circulation began introducing wind events from the north and northeast. These offshore-directed episodes had a measurable effect on the bloom's coastal concentration. Offshore winds do not destroy a bloom, they push the surface water and the organisms within it away from the coast and into deeper water. Water quality monitoring stations near Fort Myers Beach and Bonita Beach recorded declining Karenia brevis cell counts during offshore wind intervals, with some stations dropping below 100,000 cells per liter, the threshold NOAA associates with moderate bloom conditions. Rainfall associated with the frontal systems introduced additional disruption. Freshwater input reduces surface salinity and increases water column mixing. Karenia brevis tolerates a relatively wide salinity range, but it is sensitive to rapid salinity fluctuations and to vertical mixing that precipitation-driven convection produces. Heavy rainfall events in late September and early October disrupted the shallow, stable stratification layer that had allowed the bloom to maintain its dense surface concentration through the summer. When the water column mixes vertically, bloom cells are distributed across a greater depth range, reducing available light per cell and increasing the proportion of the population below the photic zone where photosynthesis is possible. Water column mixing also brought cooler, nutrient-depleted water from depth into contact with the surface layer. Diatoms and other phytoplankton species better adapted to turbulent, mixed conditions began recovering in bloom peripheral areas where cell counts had declined. The reestablishment of community diversity in those areas signaled that the environmental conditions specifically favoring Karenia brevis monoculture were eroding. The reduction in freshwater discharge from Lake Okeechobee through the Caloosahatchee River system during October contributed further to the shift. The Army Corps of Engineers had modulated release volumes in response to declining lake levels and regulatory pressure. Lower discharge volumes reduced the nutrient pulse supplementing the nearshore chemical environment since June. Without that continuous external input, the bloom's growth became increasingly dependent on ambient Gulf nutrient concentrations, a lower nutrient environment that Karenia brevis can persist in but cannot expand rapidly within. By mid-October 2018, field sampling data reflected a consistent pattern of declining cell counts across the bloom's coastal footprint. The densest concentrations had fragmented. Satellite ocean color imagery no longer showed the coherent, high biomass surface expression visible since early August. Karenia brevis populations remained detectable at low to moderate concentrations in portions of the Lee and Charlotte County nearshore zone, but the threshold concentrations associated with fish kills, aerosol hazards, and shellfish contamination were no longer being sustained. The practical consequences registered quickly. Shellfish tissue samples showed brevetoxin concentrations trending downward in several closed growing areas. Some beach advisories were lifted as cell counts fell below respiratory hazard thresholds. Charter fishing operators reported cleaner water and the return of live fish to areas ecologically vacant for weeks. The physical recovery of the water column was faster than the biological recovery of the ecosystem it moved through. Water clears. Fish populations, seagrass beds, and marine mammal numbers do not replenish on the same timescale. The shift that occurred through October was driven entirely by seasonal atmospheric change and natural oceanographic variability. None of it had been engineered. The bloom had begun with a convergence of favorable conditions. It ended when enough of those conditions reversed. What remained was a coastline measurably altered and a set of unresolved questions about how frequently those conditions would converge again. The 2018 red tide event ended not with a clear boundary, but with a gradual statistical return toward background conditions. Karenia brevis did not disappear from Florida's Gulf Coast waters when the bloom declined in October. It never does. The organism is a permanent constituent of the Gulf of Mexico ecosystem, present at low background concentrations in offshore waters throughout the year. What the 2018 event represented was not an intrusion of something foreign into a healthy system. It was an extreme expression of a process that is always, at some level, underway. That distinction matters for understanding what comes next. Historical records maintained by the Florida Fish and Wildlife Research Institute document Karenia brevis blooms along Florida's Gulf Coast, extending back to at least the 1840s, with scientifically monitored records available from the mid-20th century onward. That long record shows that harmful blooms are not new to this coastline. What the data from recent decades shows, with increasing consistency, is that the conditions amplifying bloom intensity and duration are becoming more frequently met. Sea surface temperatures in the Gulf of Mexico have risen measurably over the past several decades. Satellite-derived sea surface temperature records show a warming trend in the eastern Gulf, averaging approximately 0.1 to 0.2 degrees Celsius per decade since the 1980s. That rate of change may appear negligible in isolation. Applied across the thermal window that governs Karenia brevis growth, it represents a meaningful extension of the period each year during which water temperatures support rapid cell division. Warmer baseline temperatures also delay the autumn cooling that played a central role in weakening the 2018 bloom. A bloom that might historically have declined in September now has the thermal conditions to persist into October or November. Ocean warming interacts with a second amplifying factor through changes in stratification. As surface waters warm relative to deeper water, the density difference between the surface and subsurface layers increases. Stronger stratification reduces the vertical mixing that distributes nutrients, disrupts bloom structure, and introduces cooler water from depth. A more stratified water column is a more stable environment for a surface-concentrated bloom, one that is harder to physically disrupt and more efficient at monopolizing the available light. Nutrient loading from land-based sources represents the second major amplifying system, and it is one more directly connected to human activity than ocean warming. The nutrient pulse delivered to southwest Florida's nearshore waters through the Caloosahatchee River during the summer of 2018 was not an anomalous event. It was an acute expression of a chronic condition. Lake Okeechobee, the central reservoir of South Florida's managed water system, receives nutrient runoff from approximately 2.8 million hectares, or nearly 7 million acres of agricultural land in its watershed. Nitrogen and phosphorus accumulate in the lake over years of agricultural drainage. When lake levels rise and managed releases are required, those accumulated nutrients are discharged into connected waterways and ultimately into coastal receiving waters. The infrastructure governing those releases, the dikes, canals, and pumping stations of the Central and South Florida Flood Control Project, was engineered primarily for flood control and agricultural water management, not for coastal water quality protection. The two objectives are not easily reconciled. Holding water in the lake to reduce downstream nutrient discharge means accepting elevated flood risk for lakeshore communities and agricultural operations. Releasing water to manage lake levels means delivering nutrient loads to a coastline whose coastal ecosystem is sensitive to exactly those inputs. Projections from climate modeling studies suggest that precipitation intensity in South Florida will increase under continued warming scenarios, producing more frequent episodes of heavy rainfall and elevated runoff into the Lake Okeechobee watershed. More intense rainfall events generate proportionally greater nutrient mobilization from agricultural soils. The discharge volumes and nutrient concentrations that contributed to the 2018 bloom conditions are not a historical outlier to be managed against. They are a baseline that future conditions are likely to exceed. The communities along southwest Florida's Gulf Coast absorbed more than $1 billion in documented economic losses from a single bloom season. The ecological losses, measured in depleted fish populations, degraded seagrass beds, and reduced marine mammal numbers, carry recovery timelines measured in years to decades. Both categories of loss are relevant to how those communities and the agencies responsible for managing the ecosystem approach the next event. The 2018 red tide did not arrive without warning. The monitoring infrastructure detected it, tracked it, and described it in quantitative detail from its earliest offshore stages through its coastal peak and decline. What the science could not provide was the intervention that would have changed its trajectory. The tools for observation had outpaced the tools for prevention. What the summer of 2018 made visible, with a clarity that data summaries alone could not convey, was that the coastline's vulnerability to events of this kind is not static. It is increasing. The ocean is warmer. The watershed is more nutrient-loaded. The bloom season is longer. The question that the 2018 event left unresolved, and that subsequent seasons have only sharpened, is not whether conditions capable of producing another event of this magnitude will occur again. It is how much of the ecosystem and the human systems built around it will remain intact when they do.